Hello again. Today I'm going to talk about translation. So once DNA is transcribed from in the nucleus, the mRNA is then released out of the nucleus into the cytosol, and then translation happens when mRNA is combined with ribosome to produce necessary proteins. So translation is the synthesis of a protein from an mRNA template. So you need the recipes for translation are mRNA, large and small subunits of ribosome, tRNA, and release factor. So the first step is pre-initiation. So pre-initiation is when this enzyme, amino acid, so tRNA synthase, attaches amino acids to tRNA molecules using ATP. So basically what happens is that amino acids combine with ATP using this enzyme, forming a bond between amino acid and AMP. And this bond, after this AMP is released and the bond between amino acid and 3' end of the tRNA is made. And this end codon of the 3' end of the tRNA is CCA. It's always CCA. And it contains a very high bond energy. So this bond energy can later be used to connect amino acids together during translocation to produce polypeptide chains. And the second is initiation. Initiation is the assembly of a ribosomal complex. And ribosomal complex separates into two, small and large. Small subunit of ribosome binds to the 5' side of the mRNA. So this is, let's say, this is the cap, and this is 5', 3', and this is, you can't see very clearly on this one. This is the start codon, AUG, and this is where the translation initiates. So, and also there is also a large subunit of the ribosome, and this is where the tRNA binds. And the enzyme that associates with the AUG start codon is called methionine. And the third step is called elongation. Elongation is when consequent amino acids coding for mRNA are brought to the ribosome, which means that after this AUG and binds to the UAC tRNA that contains methionine, the second codon that follows this start codon is elongated during the translation. So, and this is A site, and this is P site, and this not shown is called E site. So this is where the tRNA exits during termination, and during elongation. Yeah. And the bond between these form, and this is called peptide bond, and the molecular structure is this as shown. Okay. So the fourth step is translocation, is when amino acids bind together through a peptide bond, growing into a polypeptide chain, and this is when this tRNA enters the E site. The enzyme, the amino acid is already connected to here, and does this when tRNA enters the E site, the tRNA just exits. So, so AUG codon starts at P site. Then the following consequent codons for the mRNA binds at the A site, and this is P site.